Hello, everyone. Today I'm going to talk about the autologous stem cell therapy for type 2 diabetes research. And my company is called a Royal Cell Biotech Corporation. Disclosure, I am the CEO of the company. Company introduction. And our company is based in California. It's an international company with research team across the Pacific Ocean. We have teams in China, in US, and we have a very unique IP stem cell technology. Basically, we use the autologous adipose-derived stem cell to convert them to embryonic stem cell without Yamanaka viral vector transfection like we're doing for iPSCs. Essentially, there's no genetic modification in achieving this conversion. And finally, we convert the EC to the pancreatic beta cell with high functionality in terms of producing insulin in vitro and in vivo. The ongoing research projects with the completed preclinical trial or animal study and initial clinical trials are in both basically from China, and the results brought great hope on stem cell therapy in both basic science and clinical research. And our current status is that we are working with the US-based CRO company in pre-IND application for US FDA for diabetes 2 therapy. And let's have an overall description of our stem cell therapy in this project. There are 5 steps we're doing right now, and then step 1 is we have the patient selected according to our inclusion and exclusion criteria, and register them in electronic medical record with plus biometrics system. Basically, we can use fingerprints to pull out the files and be very accurately manage the patient cell storage. And the clinical trial registration is for 3 years. Step 2, CMC, or the cell preparation production for the project, and autologous fat extraction for the MSC stem cell is completed and purified, and cell expansion finished, and we convert them to the EC, and then from EC induction back to the beta like cell of the pancreas, transfect them with the insulin booster vector. Those are the steps for the cell production. Then we characterize the cell, and then cell function study, and then cell stability study. I will get into details later on for those steps. These are very important steps for this project. Step 3 is we have completed all the animal studies, including diabetes animal models and pharmacokinetics, toxicology, allergy, and functionality studies on the diabetes animal models, and carcinogenic study, and also independent third-party GLP verification, QC, all those steps were finished. And step 4 is clinical trial design and prosecution with the various cells we're producing, and the trial is ongoing, and we have 3 years study for this. And step 5, which is what we're currently is doing, we are shifting the pre-IND FDA applications in US, and then we are giving this all the organization management to a CRO company here, which is also a well-known international company in the US. So we start with patient selection, registration, biometrics, and physical exam, blood test, biochemistry panels, A1C, C-peptide, etc. Inclusion should be 35 to 65 male, female, diabetes type 2 for 3 years or longer, A1C more than 8.0, consent for 3 years for the study, exclusion, drug addicts, contagious disease, cancers, severe diseases such as renal failure, and follow-up physical exams, clinical symptoms, medication dosage, such as insulin and complication data, and of course biochemistry panels too. Autologous fat extraction. So using the standard handheld suction device to prosecute the liposuction technique, and the tumescent fluid from the abdominal area of the patient, and then 5 cc of the fat is processed with collagenase and followed by 3 washes with 50 cc of buffer solution, and the mononucleated cells is isolated and ready for culture, which the technique is very similar to the ICSS time machine protocol. So next step is mononucleated cell are cultured, and morphology and cell differentiation to adipocyte and osteocytes are confirmed. Cell morphology are confirmed with spindle shape are demonstrated here, and also the CD marker CD44 positive and CD34 negative are also confirmed here, and cell differentiation to osteocytes or osteogenic differentiation and adipogenic differentiation are confirmed here. So this step is MSC to the embryonic stem cell and its verification. The isolated and cultured MSC were converted to the embryonic stem cell by proprietary technology without modification of the gene. As such, in iPSCs, vector are usually transfected with the Yamanaka factors to the cells in order to achieve the conversion. The TRA IgM marker are used to confirm the MSC conversion to the human embryonic stem cells. The whole process of this technology paved a brand new way in autologous embryonic stem cell application for the therapy of various diseases. Now, next step is phenotype and CD marker of the S cell or EC cell characterized. So the cells are cultured at flask of 90% fluency and divided 3 times, then multiple passages were repeated. I'm not going to get into the detail of this, but the cell number, quantified, morphology are confirmed. And CD markers are also confirmed with different CDs, as you can see here. And the cytoplasmic expression of the gene product are also confirmed. And even the cell passage over 50 times, the cell remains the same biomarker and characterization, meaning it's very stable at this point. Following step is the EC is differentiated into beta-like cells of pancreas. Basically, the autologous EC is differentiated into beta-like cells of the pancreas to secrete insulin in a 3-step method of induction. And the confirmation staining of the beta cell is called DTZ staining, and it's confirmed in a brownish color. So the beta cell is then undergone further viral vector transfection to increase insulin production. The clinical grade lentivirus vector were transfected to the beta cell, and then the transfected beta cell vector booster, we call it insulin booster, is about 4 times of the insulin compared to the non-transfected one. The insulin is standing in red here, is demonstrated on the right side of the figure here, right here. This work has been published actually at Stem Cell and Regenerative Medicine. And all the cell final batches are tested for the QCs by independent third-party testing center. All the pathogens, HIV, HBV, HCV, EBV, cytomegalovirus, bacteria, fungus, and mycoplasma, endotoxin, etc. Okay, so by now, let me give you a brief summary to avoid any confusion. Basically, there are 3 cells in this cell production. Okay, the first one we call it S cell. Basically, it's MSC. It's autologous fat extraction, purification, expansion, CD marker morphology, and cell differentiation are all verified. So it become the second step is called SLS, which we basically say it's an EC or embryonic stem cell. So using the MSC converted to the EC without genetic modification, and also confirmed they have the EC CD marker and also genetic product identification, and then later it was converted, was induced to the pancreas of the beta cell. And then that's also confirmed with staining with all the characters and also insulin production. And the final step is called SL-S-insulin, which is basically the beta cell with the viral vector. So the beta cell are obtained from the prior step and then co-transfected with viral vector, which is an insulin booster to increase about 4 times of the insulin. So the final stage is the beta cell with a high yield insulin production, and this way it's much more efficacious in the therapy of type 2 diabetes in the trial. So the animal study take a long time, but I'm only going to use 1 slide. The study basically on mice, rat, monkeys are all completed in a GLP lab in China. Toxicology with IV, tail vein IV infusion, hemolytic test, allergy test, carcinogenesis, vascular stimulation, skin injection, models, different route of model giving the stem cell were tested, and diabetes animal models also confirmed for the therapeutic effects on the animal models. So next is the human pilot test in progress. There are 3 arms are here. As I told you, there are 3 kind of cells. The cells started with MSC, and then beta cell obtained from the conversion, and also beta cell with the booster. So we have A, B, and C arm. With the MSC only, after treatment, same treatment, 50 million IV, 3 doses, and you got 11 A1C to start, and you end up with about 7 after treatment, and nearly half of them are efficacious, which is well known that MSC is efficacious in treatment of diabetes. We have data from this ICSS also showing that. I think it was even higher, if I remember. And then with the beta cell conversion, B arm, we have start with 11, and then we have a better outcome um, than the MSC only, and then we have a higher percentage in terms of efficacy. And the C arm with the booster with 4 times more insulin, um, we are I seen individual data, but I don't see the statistics yet. But I heard it's 80% in efficacy, and hopefully that will be our final choices. Um, that is the current status for the human clinical trial. Okay, let's have a summary of the study. Autologous adipocytes are very easily obtained, and MSC are easily isolated and cultured and purified and characterized. Conversion of autologous MSC to EC without genetic modification paved a brand new way for autologous cell therapy application, which is much more acceptable than iPSC, which you have to use viral vector for the Yamanaka factors. This conversion technique has made future stem cell therapy much easier and available without any ethical issues. So we can basically obtain somebody's autologous embryonic stem cell to convert to any cell type for the purpose of the treatment. The CMC protocol and data for the cell production are very reproducible, which will be further tested by the CRO company guided GLP lab in the US. Complete animal study and pre-human trial are ongoing in China and will be repeated in US. So the setting of the location of the beta cell or beta-like cell and is not known right now, and whether the secretion of the insulin is responsive to the fluctuation of the glucose level in vivo or in vitro are currently under study, and the extended duration of the insulin secretion is yet to be determined. So current efficacy lasts about 3 years. Can we do it one treatment lasts for 5-10 years or even permanently? We don't have the data right now. Now let's talk about the potential pitfall in this project. Lack of sustained production in insulin or overproduction of the insulin. And so we have 3 years, but we don't know how to extend the time, and also we don't whether it will overproduce and cause hypoglycemia, which never happened, but it's possible. Okay, and then mutation of the cell in vitro expansion. So anytime you expand the cell in a laboratory setting, you are running a theoretical risk of mutation and maybe causing cancerous cells. An abnormal cell setting and growth in an unknown location. For example, the cell didn't sit in the pancreas, maybe sitting in the brain or somewhere else, and it's growing in mass, and we don't know. Factors influence the cell insulin production other than glucose stimulation, such as hormone, estrogen, because the promoter is the estrogen promoter, and it's not unknown. Okay, and then any other unknown outcome we have to watch with time. And so far, none of the above has been observed, but that doesn't mean it will not happen. So let's talk about the pathway for the US FDA and clinical trials. the pre-FDA meetings and review of all current data and repeat some of the experiments and address those pitfalls and related issues. We already have prior feedback from CRO and unofficial channels with FDA, and we are hopeful that our project is going to go in approval process. The CMC of the cell production under the U.S. CRO company or companies can give us a safe, reproducible results and stability study finished, cancer risk assessed, and long-term toxicology completed. And then we can go to clinical trial, hopefully around 3 years. Okay, that's come to the end of my discussion. And I really appreciate everybody spend time to listen to my presentation. The main purpose of this presentation is to get feedback and instruction from the expert panels and audience here. And feel free to contact me. And my cell number here, the last line here, 626-688-5968. And if you want to write me, my email is drzenmd@gmail.com. DRZENGMD@gmail.com. Really appreciate everybody. Thank you very much.